Thanks, everyone, and welcome to this special I3 session, Mind the Generational Gap, the Next Generation of Embryologists. So there might be some misconceptions about some of the older people that are doing IVF and some of the younger people, often referred to as Gen Z, right, Huey?
Yeah, and millennials, I guess we should throw them in there too. I had to find out the age range between the two.
Well, through most of this, we're going to dispense with the long bios and the contact information, but go to ivfmeeting.com and you can find that information there. And we encourage you to make sure that you ask your questions, not in the chat section, but in the Q&A section, and then we'll be able to address those. I'm joined here by Huynh. He's dedicated over 25 years to education as an adjunct instructor in anatomy and physiology for healthcare students at several local colleges in Pennsylvania. He's served on educational boards and as a guest lecturer in IVF and human physiology. We're also joined by Savannah Palmer, a research postgraduate student at Imperial College in London within the Institute of Reproductive Biology. Her background is focused on embryology and microbiology, resulting in a fascinating PhD project that combines these two fields. We also have Toyin, who upon completion of her bachelor's went on to pursue a postgraduate Master of Science degree in human clinic embryology and assisted conception at the University of Dundee in Scotland. After successfully finishing her master's degree, she was driven to become an embryologist, and she joined the embryology team at Repromed Galway and is currently undertaking a traineeship.
Okay, thanks, Kimball. Let me tell you a little about this guy, a special friend of mine, actually. Dr. Kimball is the Chief Science Officer for IV Fertility and oversees several IVF laboratories in the US. He's an excellent and very engaging lecturer and received his PhD in animal physiology at Colorado State University, where he learned how to fly fish as well, I assume, for when he's on the lab. He did his postdoc work at Salk Institute for Biological Studies in California. And at the Institute, Dr. Pomeroy worked with Dr. Glenn Evans on the Human Genome Project and did work in the production of transgenic animals. And you read more about his career and all his accolades in the IVF meeting website. But beyond all of those things, like I said, he's a special friend, and I think his best attribute to the field of art is the humble and genuine nature in which he presents his intelligence and expertise. So I recommend every Gen Z and millennial to really bombard him with emails because he will actually take his time out of his day to give you a very thoughtful response. So we are also fortunate to have an expert in human resources who will help us navigate tonight's session. Angela Regan, she has been a HR director for Care Fertility Group in the UK for the last 20 years. She graduated in law before completing a postgraduate graduation degree in human resources management. Angela's role is to continue to develop and implement a people strategy that considers and meets the needs of a talented workforce. Thank you, Angela, for being here. I just think your perspective in recruiting and nurturing young embryologists will be of strong interest to a lot of the people here. So let's go ahead and welcome our first speaker, Fran Farlie. She's an I3 ambassador who's been working in the art field for a couple of years and completed her master's degree in 2020 at the University of Dundee in Scotland. She was inspired by an earlier I3 webinar on green IVF. So Fran has worked to encourage sustainable practice in reproductive medicine and has been a speaker at the ARCS Summer Symposium and will present at Fertility in 2023 next year. So I welcome Fran also. I'm looking forward to hearing your talk. I used to teach environmental science, so I really will be engaged to see how your thoughts on implementing sustainable technology and strategies in IVF.
Hi everyone, I'm Fran Farlie. I'm meant to be speaking to you today about green IVF, and I'm so sorry I can't be there. I've finally taken some annual leave. I've got the bag packed. I'm heading to the airport in hope for some warmer weather. But being part of this younger generation, we know how to use technology, and I can't have you miss out on the green message. So I will be with you tonight virtually. Don't worry though, if you have any questions, you can get through to me on Twitter and I'll get back to you as soon as I can. I hope you have an amazing evening and a great Q&A at the end. Bye!
I'm here to talk to you about green IVF, a term coined by the I3 group. I have nothing to declare. I'm an ambassador of these three things at the bottom. Climate for Health is advocating for sustainability in healthcare, mostly in America. And my green lab is more for research labs, but there's definitely information on there that we can apply to the ARP field. They're free online training programs, but with great resources, so it's worth checking out. So today I'm going to talk about why we need to be more green, what things we can do to be more green, and then how we can ensure sustainability in the future. Now, normally I would bring Kermit with me to tell you he was wrong and it is easy being green, but he started in 1955 and tonight's all about generations. So instead I've brought the UK's green mascot from my generation, which is Dipsy from the Teletubbies. So why do we need to be more green? We know about the rising temperatures, polar bears, turtles, but why specifically in terms of reproduction? Now, evidence is still very preliminary and cause and effects aren't entirely clear, due to the complexity of reproductive science. But I thought I'd cover a few issues that have come up so far. So the first two is things like increased temperatures and air pollutions are being linked with adverse birth outcomes. Systematic reviews, and quite large systematic reviews, are starting to show, particularly in asthmatic mothers, being exposed to air pollution can increase the risk of preterm birth by 52%. Similarly, air pollution, a high exposure to air pollution in the third trimester can increase stillbirth by as much as 42%. So there's something we need to be aware of. Another factor is microplastics, and more studies are starting to come out now. But microplastics are endocrine disrupting chemicals, so they mimic our endogenous hormones and wreak havoc in the body. Studies are starting to find microplastics in our reproductive tissues, so testes, the placenta, both on the maternal and fetal side, and even the umbilical cord is finding microplastics. They've also been linked with poorer semen quality and causing problems with ovarian fibrosis and apoptosis of granulosa cells. So we've got potential male factor, female factors, and developmental problems that could be linked with microplastics. Finally, we have microborne diseases. As temperatures increase, habitats and behaviors of disease-carrying insects changes. In America, the CDC reported cases of vector-borne diseases almost tripled between 2004 and 2016. And what we're interested in is things like Zika and malaria, and it's either going to lead to increased costs for testing for these things for our patients if they're going through fertility treatment, or delaying their treatment if they've traveled to countries like this. So as healthcare professionals, we need to be wary of climate change for our patients' treatment. So what can we do to be more green? Hopefully I've convinced you that we should be looking to be more green. And I'm not suggesting that we'll be able to reduce the planet's degrees by a few degrees, or planet's temperature by a few degrees, but we can help to not cause any further damage. So in order to do this, we need to understand where our carbon footprint is coming from, but also look for sustainable solutions that are not going to impact our patients' quality of care. So where do our emissions come from? Well, 71% come from our supply chain. And that's because it's not just how we use products in the clinic and how we dispose of them, but everything that comes before that. So mining to get our raw materials, the manufacturing process, which is predominantly run using fossil fuels, so that's contributing to air pollution, and then transport both within manufacturing and to the clinics, all contributing to the emissions in healthcare. So we need to be optimizing our use of resources, steering away from this culture of excess and denormalizing disposability. We need to think about this waste hierarchy pyramid. And if you haven't seen it, at the top, it's having avoiding using resources. If we can't avoid it, can we reduce it? If we can't reduce it, can we reuse it? And then at the bottom of the pyramid is just throwing it in landfill. So an example of this would be using serological pipettes to measure semen volume. Now, instead of using serological pipettes, that is the plastic and comes in another plastic wrapper, we can be using weighing scales. That would be both more accurate for your semen volume. You're not losing sample, which what serological pipettes would do, but you're also reducing the plastic use in the lab. And it'll be financially beneficial in the long run because you're not constantly paying for more and more serological pipettes. So I know lots of people have tuned in today all across the world, and we need to be using our global purchasing power to encourage suppliers to act more environmentally conscious. So companies like Johnson & Johnson and Philips have already set targets and said that they're going to be carbon neutral, but we need our IVF suppliers to look for more sustainable options. And perhaps some sustainable options that are out there are maybe not as conventional as you might think. So if you are tuning in from Australia and New Zealand, although I think it might be really early given the time zones, you might be able to attest to the fact that sheep's wool is currently being used. So it's replacing these styrofoam boxes. It's 100% discarded wool that's been sterilized and wrapped in compostable insulation. It's reusable, recyclable, and biodegradable, and it takes up much less space and is easier to manage. But unfortunately, changing products once they're available to purchase is a lengthy process because they need to be approved and relicensed or something like that. So it's getting suppliers to change is going to be more of a long-term goal. But if we encourage our suppliers to act more sustainably now, then they can start that process in changing their products. So please email your representatives, ask them what are they doing to be more sustainable, but also what can you do in the clinic that once you've used their products, how can you dispose of them more sustainably? So that's the long-term goal, but what can we do in the clinic for the short term? Well, there's a few suggestions. Reusable hats is one of them. And if you haven't used them already, it's definitely worth switching to. In the UK alone, in healthcare, every 4 seconds, a disposable hat is being thrown away, which is a crazy number. There is currently no evidence to say that reusable hats increases the risk of infection or contamination. So as long as you have a good cleaning SOP, then there really is no reason why you can't make the change. Ice packs is another one for our media being delivered. If you can avoid throwing away the ice packs and extending their lifespan, then you would be reducing your carbon footprint. So trying to partner with schools or sports facilities, or even speaking with your local council or donating to charity shops, if you can find an alternative to the bin, then you'll be reducing your carbon footprint. Paper is another one, once told to me being the biggest contaminant in the lab. We want to increase digitization, move away from paper, but if we really have to use it, we want to be printing double-sided and be using paper from a recycled source. Recycled paper compared to virgin paper uses 50% less energy, and therefore you'd be reducing your carbon footprint. Energy is another big one. The cost of living, everything's becoming more and more expensive. We want to be swapping to LED lights if we can, or if you haven't already. Installing sensors, motion sensors, particularly in those sort of small intermediate rooms that no one's really in there for a long time, but you have to move in and out. If you have a sensor, it will automatically turn off when nobody's around, so you'll be saving energy. But also outlet timers in the lab. If you've got equipment that's running, especially heated equipment that's constantly running, you can use an outlet timer. So at the end of the day, it will automatically switch off. And then in the morning, you can set a time for it to turn on again. And so it will reach the temperature that you want before you've even reached the building, but it's not been on all night. And then that can help reduce your energy costs by at least 50%. Laundry is another one. Specifically, if you're a clinic that uses a third-party company to do your laundry, there can be a lot of plastic bags involved. And that's both for taking laundry away and delivering again. If you can find sustainable solutions or alternatives, then that's something worth doing. And then finally, water. As embryologists or as scientists in the field, we're always washing our hands in and out of the lab. You can install aerators into taps, which can reduce your water consumption by up to 50%. So these are just little short-term things that I think we can all be doing ourselves. So what can we do looking forward to ensure sustainability of healthcare? Well, in the UK, we're a little bit ahead on this. We've got the first piece of national legislation in the world to address healthcare sector's response to climate change. As part of this, they've set goals by a certain year, so 2040 to be carbon neutral for direct emissions, so those are the things they can control on site, and 2045 for indirect emissions, and that's things like the supply chain. As part of this, the NHS have made mandatory green plans for each trust, and this is something which I think we can pay attention to. So there is a document online that's easily accessible that outlines how to write a green plan, and it covers your sustainable targets, so your resource consumption, your paper, energy, water, that sort of thing. Transport, so trying to make greener commutes, so adopting a cycle scheme or a car sharing network. It's trying to improve your commutes. There's national focus campaigns, so we've just had Environmental Week, but also Earth Hour or Recycling Week, and then staff training. We already do like the handling liquid nitrogen training or the equality and diversity training. Why not have a 10-minute module that covers how to be sustainable in the clinics? It also outlines how to report your progress with audits and reports. And then one of the big ones, which is something that I've done in my own clinic, is forming a green committee. So we suggest ideas, we have meetings every 3 or 4 months, we discuss things we've implemented, things we want to implement, and then we action those things to be more sustainable. So this is something you can do in your own place of work, or if there is a trust with multiple clinics, you can have a joint green committee. And also there's a potential interest in maybe we set up an international green committee and really tackle the issues in healthcare. But this is where our policymakers and societies really need to step in and enforce sustainability. So ASRM have actually made their red journal green by digitizing it to lower their carbon footprint. And they made a rough calculation that the fertility and sterility subscriptions, once moved completely online instead of in print, would save 2,200 trees yearly. But this really is only scratching the surface of the things that they can do to improve the greenness of our field. The HFEA, in their 30 years of regulating, so this is in the UK, came up with a statement that said, although our statutory framework is old, where we find unethical practices, will not hesitate to speak out. And I would argue that the culture of normalising disposability is unethical, since we know that the impact of climate change is adversely affecting our reproductive health. So we need to advocate for guidelines to make it easier for us to make the sustainable switches. I've spoken with someone from the HFEA who agreed that there is a lack of sustainability in healthcare when she once worked in a clinic. And she suggested that if a policy document was to be made or a best practice document, then they would be interested in sharing that through their clinic focus, which is like a monthly newsletter that goes to all staff. So this is something that I am starting to work on, trying to come up with a document that can be sent out to everyone. We already have the Cairo consensus for air quality and VOCs. We have the WHO lab manual for seed analysis. Why not have something to set the standard for our environmental impact? So everyone plays their part. They have a part to play. Individual actions is great and we need it, but we need to pair that with some systemic change from our suppliers, from our governing bodies, and then we'll be able to make sustainable solutions easily accessible for everyone and make it become the norm. So while I was preparing for this presentation, I was reading a book, and the very last page of the book by Dr. Chris MacDonald covered generations, and I thought it was just perfect for this webinar. So I'll just finish on this. So it's a book from the Operation Sustainable Human, and it finished with, we could be a generation that not only wanted change, but were willing to change. A generation not only against the climate crisis, but proactively for the available solutions. We don't have to drag the past with us, we can break the mold. So thank you, Ivy3, for having me. Although I can't be there in person to answer your amazing questions, feel free to contact me. You'll find me on Twitter and I'll get back to you as soon as I can.
Thank you so much, Fran. We really appreciated that. I'd like to find out if any of the panelists here have a recommendation. I know that I've always been bothered about the tons and tons of styrofoam I've thrown away in my lifetime. I feel I could build a mansion or a small city with all that styrofoam, although it might be hazardous if it caught flame. But what can we do with it? That's something simple that we can do. And you think there'd be a solution? Do any of the panelists have a recommendation of what we can do with styrofoam that all of our media comes in?
I can butt in here. Am I allowed to?
Absolutely, Chloe.
Okay, right. I mean, what do we even use styrofoam for? It's like basically protecting the stuff that we're actually carrying. There are plenty of materials that are way more like sustainable than styrofoam that can do that job. The only question is whether or not we can scale them enough to make them cheap, or like whether we can like essentially find them cheap enough. I'm not a material scientist, but I'm sure that there is a material. I think it's just a habit at this point that we're using styrofoam, and the fact that we've got economies of scale.
Maybe if we talked a little bit more to our manufacturers, we could get them to set up a way that we can recycle it, or recycle it locally so that we don't have to ship it back to them.
Yeah, like at least in my clinic, what we're trying to, we specifically speak to our suppliers. So for example, with blood test kit boxes, we specifically speak to our suppliers about making them more environmentally friendly, making sure that like the box that everything comes in is recyclable. So like it's not something that you get by default, but if you ask, it's quite often the case that they can offer something.
Thank you very much, Chloe, and I'm grateful for Fran for presenting that information. Now it's my pleasure to introduce Radhika Kakulavarapu. She is a young, eager embryologist who's currently dabbling in research regarding the morula stage embryo and irregularities in compaction. She's pursuing her PhD in Norway, where she works with a large interdisciplinary team that investigates the use of explainable AI. That's not artificial insemination, that's artificial intelligence within the IVF clinic. So you can read more on the website, but I'll turn it over to you, Radhika.
Great, thank you. Can everyone hear me and everything? I hope so. But anyway, hello everyone. I am super grateful to have this opportunity to basically talk about my life and my experiences so far. That's definitely a first for me, so thank you, Giles, for reaching out. So compared to some, my list of experiences is not very long, but much like my main research interest, the morula, I find that I'm constantly changing and evolving in a pattern unbeknownst to anyone but myself, essentially. So here I intend to show you glimpses into my career in embryology so far and what I have learned in the process. So without further ado, here is moving like a morula, a millennial experience. I'm primarily affiliated with Oslo Metropolitan University, colloquially called OsloMet, and I have no conflicts of interest to declare. So when I was thinking about how to start this talk, I simply decided to start at the very beginning and give you guys a quick rundown of what and where makes me me. So I was born in a small town in the south of India where I didn't remain very long at all. My parents and I then moved to Hong Kong when I was 4, and this became the place of my childhood, where my sister was born and where we spent over a decade. Then we moved to Dubai, where I finished up high school and where my family lives now. So I guess that's home base for me. I then continued on to do my undergraduate degree at the University of Toronto in Canada, and decided to specialize further in clinical embryology, which led me to do my master's program at Monash University in Melbourne. So after all of this, I took up my first job as a junior embryologist in Osaka, Japan. I was there for 1 great year before moving to Cebu in the Philippines, where my role as an embryologist expanded a little bit and differently. So after another year of there, COVID hit, and now I find myself in Oslo, Norway, where I am pursuing my PhD. So obviously I have moved around quite a bit, but the real exciting embryology stuff happens here in Japan, where I talk about the extensive clinical role or the clinical skills that I gained there. In Cebu, where I zone in on a more corporate or business side of the industry. And now finally in Oslo, where I'd like to consider myself as more of an academic embryologist. So first step, Osaka. This was a very exciting year for me. This is fresh out of my education. I was primed and ready to take on anything and everything. This year was my first IVF family. And believe it or not, everyone that you see on the bottom of there is at least a decade older than me. The skincare over there is just fabulous. So if you haven't tapped into that, please do. But in terms of generation gap, there was that aplenty. That was obviously also compounded by the fact that there was a pretty obvious cultural gap. As the protagonist of this presentation, yes, it was difficult to transition for me to learn the Japanese language, the work culture, and train as an embryologist on top of that. But I'd also like to point out that it was probably equally, if not more difficult, for the rest of my team as well. Explaining procedures was significantly harder. Explaining cultural experiences or expectations was harder. And the intricacies of working within such a large hospital was, in retrospect, quite a task indeed. So let's keep that in mind while I go along. But what stood out for me? So among 1,000 different things, I was confronted with something that maybe our more experienced embryologists might be familiar with, a mouthpiece. It was the one that they gave me was very cute. It had a cat with heart eyes on it, and it had my name on Japanese. It was adorable. But for those that don't know, this contraption that you see here is what I'm talking about. Let me just get my pointer out. This over here is a mouthpiece where this little end goes into your mouth, and then there's a long plastic tube, and on this end is a glass pipette, and you use this to essentially handle oocytes and embryos and do all sorts of processes in the lab. So I wanted to bring this up in particular because here a fresh out of college trainee embryologist, i.e. me, is faced with something completely unexpected, something that hadn't even been mentioned to me as a possibility in my education just a couple months prior. So in my mind at the time, I was immediately very defensive, and I thought, okay, I'm being trained for something obsolete or something in the past. Now, I'm not sure about you guys, but my mom absolutely hates it when I ask her why constantly. So when I asked my supervisors, naze or doshite, which means why or how, this was very much discouraged, or it was considered more disrespectful because it was the general practice of the clinic at that point. And more than that, it interrupted what was colloquially called the samurai method, which is essentially the Japanese way of saying practice makes perfect. So eventually I did complete the necessary levels, and now I'm proud to say that this is me brandishing my mouthpiece, metaphorically. So I'm primed and ready with that technology at least. But all in all, I did learn a lot about older practices in our field that maybe aren't explicitly mentioned in current education. And though it came slowly, I started to break preconceived notions on how things should be and started forming ideas of how things could be instead. So I'm going to end on my Japanese experience over here with a local proverb that's stuck with me over the years. The bamboo that bends is stronger than the oak who resists. So essentially, flexibility and adaptability, at least in my case, really drove me forward as an embryologist. So after a year, I took up an offer to work with a new clinic in Cebu, where I was involved in setting up a lab from scratch. So this meant designing floor plans, contacting vendors, ordering equipment, talking to fire safety people from the building, laying down gas supply lines, just all the things that I had only even thought about in those hypothetical assignments that they make you do in your programs, if anyone can relate. So this is finally an assignment that was actually really being put to use. So that was great. But this was surprisingly difficult to wrap my head around for me. But I did the best I could. And from my experience in Japan, because things were generally older, I was able to come up with creative cost-cutting solutions and discuss this with owners and clinicians, where it was generally, or at least to me, it felt like more of a team effort than clinicians being separate and embryologists being separate and nurses being separate. So that was really great in my opinion. So we were also building our patient base quite slowly, and I was involved in a lot of consultations, which is relatively new to me because again, in Japan, everything was quite quality controlled. Everything had to be a certain way, and that worked very well in their clinic. But here was a complete 180. Patient consultations very often ended up just drinking tea in the office with the doctor, and that was very different for me, but it was beautiful. I thought it was fun, and the patients had a good time. So everyone seemed to be benefiting in some way. So as we did this, we started our treatments. Things were growing slowly, and it was great. But then, as a largely clinical embryologist, or at least that was my idea of it, I had this overwhelming thought as the more business side of things and the more corporate side of things slowly dominated my everyday life. I started thinking, am I still an embryologist? So after talking to a few people in the field, I know I'm not alone in thinking this way. Sometimes it might be a slow week in the lab, or maybe you find yourself with a mountain of paperwork while you'd rather be in the lab. So I don't know, maybe some lab managers can relate to this sentiment, maybe. Needless to say, I had withdrawal symptoms from the busy lab experience in Japan. And though it took me a while to get there, I now see this as an expansion to my current skills. And so although the samurai method will always be accessible to me, this experience in particular in Cebu gave me skills in autonomy, decisiveness, leadership, and a general interactability that will only drive me forward, I believe. But hold on to your butts. Here comes another local proverb. This one really I loved. The sleeping shrimp is carried away by the current. Essentially, again, the way I interpret it is that proactivity and involvement in every facet of the embryologist role, whether clinical, whether corporate, it definitely, at least for me, made me a much better embryologist to understand why we were doing the things we did and what, at least from the outside, it came across as. So to the last and current stop on my journey, Oslo. Unfortunately, the clinic in the Philippines could not survive financially after COVID-19 hit. And so this, one, left me out of a job, and two, desperate to use all the skills I had picked up along the way in a very short amount of time. So now I find myself as a clinical embryologist on the outside looking in through the view of academia, and especially with a unique perspective. So using my clinical background and newly attained corporate skills, I was ready to bridge gaps, not only in my own knowledge, but maybe transfer them to embryologists that could also benefit from the same. So I am now, as Kim mentioned, I am now part of an interdisciplinary team that researches the use of AI methodologies within reproductive science. So now I have this amazing opportunity to dip my toe into AI and be part of the innovation sprouting within this field. Some of my team is here. This is Erwan, my main supervisor, my co-supervisor Metta, Asset Eshrei and my first abstract. It's all super exciting. But speaking of my research, I should probably touch upon what my research actually is. So like I mentioned at the beginning, my interests lay within the morula. But to focus in a little bit more, I am looking at a phenomenon termed cell exclusion, which some may recognize as partial compaction or lazy cells. And what we see over here is a very clearly delineated cell that remains outside of the morula or start blastocyst stage. There is very little literature looking into this. It's relatively newly defined. So what we're trying to do here is characterize this anomaly a bit better. And initially, we are looking into some non-invasive morphokinetic characterizations, but we have also started pilot testing to look into adhesion proteins to try and explain why this happens or how this happens. Yeah, so that's what I'm doing. But essentially, apart from cell exclusion, the overall idea or the scope of this project is to essentially use AI goggles and to see whether we can look at the morula again with a fresh perspective and to fill the gap in our current knowledge that we see. And I know that unpacking the black box is a very common term in AI. So I'd like to consider this as unpacking the morula a little bit. Yeah, I won't end this proverb. I won't end this this experience with a proverb because my journey in Norway is not over yet. But my current experience as an academic embryologist has definitely opened my eyes in unexpected ways, and in ways that I hope will cross generational and cultural gaps with ease. So now to make sense of my experience, as waves of younger embryologists enter the workforce, I would like to take this opportunity to dispel some incorrect stereotypes, in my opinion. First, that job hopping makes you disloyal. Yes, I am guilty of job hopping, but I am starting to learn from my fellow data scientists, and correct me if I'm wrong, but more data isn't always better data. Your algorithm will be better with different types of data, generally creating a wealth of experience that I believe younger people will have that may be more valuable than you think. Next, that we have a short attention span. Like the proverbs that I've touched upon, I find that there might be a bigger impact with fewer words. More than that, though, I believe that this may be a reflection of supreme adaptability when the situation demands it and doing more within a given time frame. And like my embryos that have cell exclusion, as you can see over here, maybe just imbibing what actually makes sense and leaving behind the bad vibes or the bad cells, maybe. Next, preoccupied with social media. I believe we can dispel this because with greater access to social media, we have such an expanded reach, much more accessibility towards patients and the services that require them. And lastly, addicted to technology. So as I briefly mentioned, the crossing of fields, especially within the realms of AI and fertility, have grown so much that this intersection may now be a field within itself. But being this bridge is important to be the challengers that exist, to be the challengers that challenge existing norms is the only way to evolve, in my opinion. So in the same way that I annoyed my Japanese supervisors and my mom by asking why, I invite everyone to do the same. We should all be asking why, and we should all be driving something forward instead. So thank you so much for listening. If you are at all interested in talking to me about our experiences, about research, please feel free to shoot me an email or check out the website for more details. Thank you so much.
Radhika, that was awesome. I actually love your thoughts on mouth pipetting, the Japanese samurai style. I think that's the thing that would pull me away from my generation and support your generation, because I feel the same way about mouth pipette. I think that would have been probably a stumbling block for me to move into IVF, because I'm like, there's no way I can do this, and I still don't think I could do it if I jumped back in the lab. So I appreciate
that's so interesting to hear.
Yeah. So I think that's not a generational thing at all. That's just They're just wrong. No, I'm joking. But I do want to ask you one question about your thoughts on AI. How do you feel that AI technologies can help this the younger generation, Gen Z, millennials, in potentially managing stress or liability or the risk that you guys encounter more so that actually we all encounter more in IVF lab?
Oh, well, that's definitely an interesting question. In my limited experience with the few younger embryologists that do handle larger amounts of data, I find that we tend to become more reliant on these objective measures and handling those little things here and there. So I think that AI can provide certain cutoffs or certain limits, but I think where the problem really lies is who's creating these limits. That's where we haven't gotten yet. So while I think the younger generation do tend to trust computer-based methods, or do tend to lean towards more objective methods of measurements, I don't think any of those things have truly been tested against any safety methods yet. But I mean, it's all a matter of time, I think. It's all developing, so we'll get there eventually.
Yeah, that's great. And one quick question also, from a cultural perspective, since you showed how you traveled everywhere, I think that's invaluable, you know, as an embryologist to your patients and working with staff. Do you feel like that has been more of a challenge than doing generational issues? You know what I mean?
Yeah, so internationally, yeah, I think in general, in all the different countries, yeah, generational gap has always been present, but I think, like I mentioned with the whole proactive thing, if you make it a problem, it's going to be a problem. Yeah. So if you reach out and you try to find more similarities between that generation, because they do exist, whether or not we want to see it, they do exist. It's just whether we want to pull out our hand and be like, okay, you know what, let's discuss this. Let's talk about it. How can we reach a compromise? Yeah. So even if those differences do exist, there's always a way forward. And we need to move towards that, I think.
Awesome. Thanks so much for your talk. It was really engaging. I appreciate it. Especially about mouth petting, of course. Right?
I thought that would be an interesting thing to bring up.
No, it was great. It was great. So thank you again. I'd like to introduce the next speaker, Chloe. She's a second-year PhD student working between University of College London and Apresticity. Her work focuses on development of computational techniques to visualize and assess three-dimensional cell arrangement in clinical embryos. I think this is going to be an amazing talk. I look forward to this personally. I think this is where AI and your work here can potentially have a great impact on the future of IVF. And this is where Gen Z and millennial generations will have a great impact than other generations that are not as familiar with this new technology that's emerging. So thank you.
Okay, cool. I'm glad that Giles told me that I shouldn't have that much branding on these slides, because normally I'm made to use the company template, and that basically means that I can afford to be slightly more creative with my slides. So I'm Chloe. This is basically my presentation. That's me. These are the people I work with on a day-to-day basis. My PhD supervisor at Apresticity is Christina. These are some people who I also happen to work with. And this is my supervisor, Francisco, at UCL. And also these are some people who I couldn't get permission to use their photos quick enough. And essentially what I'm going to be talking about is the research that I'm doing, because the brief that we were given was really vague. So I guess that we're all going to do like mildly different presentations. So my work, as was previously said, basically focuses on the idea of converting time-lapse images and time-lapse videos into 3D time-lapses. And you might be wondering, why are we doing this? And the answer is because we want to look at cell arrangement. And looking in terms of cell arrangement in embryology, even from the full cell stage, the organization, the spatial organization of cells in an embryo is going to have an impact on the blastulation potential and potentially other aspects of like clinical outcomes. So for example, we know, and this has been shown by many studies, that at the full cell stage, the tetrahedral embryo is way more likely to blastulate than a planar configuration. And you can see in the corner that one of the works was from our group. And this shouldn't be too surprising to anyone, because like when you look at it from the biological side, like these embryos are stem cells, they need to figure out what they want to become, and in order to do that, they need to talk. How do they talk? They touch each other, send some chemicals, and the more communication, the better, as the theory goes. And this kind of shows in all of the like I guess we could say like non-clinical research, so basic science research that has gone into embryology. So for example, looking at embryos under confocal microscopy and imaging like the position of cells, figuring out what signaling's going on, figuring out like how certain proteins are distributed, and generally just like seeing whether or not you get like essentially function from form. However, in the clinical setting, the picture is quite different, and you end up with something significantly less sexy than the confocal images on the previous slides, namely these Hoffman modulation contrast microscopy images on the beautiful embryoscope. And this is quite a shame, because a sad consequence of this is that the study of pretty much anything more complex than 4-cell embryos in terms of structure has kind of been the preserve of academic labs as opposed to the IVF clinic. And on the clinical side, we basically resort to like famously vague terms like the Gardner scales blastocyst contains many cells that are tightly packed together, or like loosely grouped, or loosely bound. And these words, though they make like qualitative meaning, it's been shown that like different embryologists will have different ideas as to how these work. And as Radhika did mention previously, as Gen Z, we like to look at things more quantitatively. So yeah, that's just a little meme. Okay. So the vision, just generally speaking, is to go from these like readily available focal stacks that you can get from clinics with time-lapse incubators, with 3D reconstruction, turn them into like a 3D image. And then through data visualization, we can like improve the patient experience by like bringing the patient in, explaining what their embryos are actually doing and stuff like that. From biology, or like combining this with biology, we can come up with explanations for like some of the mechanisms in like really, really early embryogenesis that wouldn't be possible without like huge volumes of data. And obviously with machine learning, looking at creating more biologically founded or biologically grounded artificial intelligence methods for selecting embryos. So I'm saying something like we're looking more than just pixels of an image in and prediction of live birth or not out, or probability of live birth out, but instead looking at potentially a more biologically principled way and saying, well, the cell arrangement or these cells have been in contact, and this cell has kind of been like hanging around on the edges, and maybe we can like even see whether or not like different like neighborhood profiles lead to, say, exclusion from compaction. So that's the rough vision, but where are we now? Essentially, where I'm at now is this idea of going from focal stacks to 3D models. And long story short, and you can read more about it in our estuary abstract from last year, or this year, from this year, yes. Effectively we take the focal stack, we then standardize it, and then we do cell detection on that, and then we generate a 3D model. This is like pretty technical, so I hope that this is like a rough, like a decent overview. I am just going to like briefly touch on, given the amount of time that we have, on the data standardization bit, because I think that is something that that's quite underappreciated, and like you don't really see this unless you're like working on the AI side. So effectively, the long and short of it is that AIs do enjoy things to be consistent, the data input to be consistent, and everything to be nice and standardized, a bit like national regulators. So effectively, we want most of our focal stacks, so these like blue things over here, and I am going to do a Radhika and whip out my laser pointer. These blue things represent focal planes, and ideally we want our focal planes to align perfectly with the embryo that we're imaging. However, sometimes your embryo can be totally misaligned with your focal plane. So it basically lives in the bottom half of your focal stack, and the top half is just blurry nonsense. Sometimes you can, for some reason, miss out the edge planes, which means that certain parts of the embryo are missing. And sometimes some people just set or some clinics just set the distance between the focal stacks to be way larger. And that also throws off the AI if we just shove it in and hope for the best. And essentially what this data standardization method that we developed was, essentially we take the focal stack and we use a artificial intelligence method to generate focal planes that would not otherwise be seen. So as you can see here, just going from top left to bottom right, you see like us going from like the -75 micron plane to the +75 micron plane. Unfortunately, if you do like some counting, you see that like the center of the embryo, it occurs somewhere over here, when ideally it should align with the center of the focal stack, which would be somewhere over here. So what if we just yeeted these two and then added two computer generated ones? And this is basically what our method does. And yeah, it's now just perfectly balanced, which is what our AI models want. And it's not just like for aesthetic purposes, you actually see like increases in the performance of AI models. So this is a very basic mask-R CNN model. You see improvements in the actual performance of the models based upon whether or not their data has been standardized. So you can see a 5% increase in accuracy and let's do some quick math, 12% absolute increase in the DICE score, which is a measure of like how well the cell has been detected. And then the other work we've been doing is basically looking at how we can bring these 3D focal stacks to the clinic. So one of the things, so in the center, you can see like a demo of an app that we've been building that allows you to see the time lapse of your embryo as it happens in 3 dimensions, which is pretty fun. On the right-hand side, you've got like a little gimmick, which I recorded with Radhika, where we've basically got this augmented reality embryo that you can walk around, play a bit, pretty fun. It runs on your phone. And then on the left, you've got some of the most recent work, which is essentially characterizing networks of cell contacts within these 3D models of embryos to better understand function. And essentially the goal of this would be to like discover neighborhoods, or like to discover configurations of cells, or like different types of configurations of cell present in not just 4-cell stage, but 8-cell, 16-cell. So yeah, that's what we're doing on this. And that's essentially the crux of my PhD. But I'd like to use the last few minutes of this talk to talk about another thing that I've been involved with, and this is these things. So it's essentially this art project that's been funded by UCL and its Public Engagement Fund, where we've been looking at LGBTQ plus experiences with family building and fertility care. And this is going to be shown at an exhibition in Camberwell in January 2023 for anyone hanging around London. But and it is an art piece. So it's in collaboration with Iris from the University of Arts London. And essentially what we did was it's a reasonably small cohort where we basically got a bunch of people to who all identify as queer to come together and work on textile pieces around essentially what they find or like what they see family building to be and their experiences, if they have any, with the fertility industry. Because obviously the fertility industry kind of like is known for catering to a very like heterosexual and potentially even affluent portion of the population. So what we did find though were a couple of interesting things. So I've got a summary of our findings on like 2 slides. The first one is like attitudes of like queer people towards family building, which might not be like the traditional, like I guess, cis heteronormative way of looking at families. So obviously, to start off with, one of the main themes that we identified within these respondents or like participants was the idea of creating support networks, that family is essentially an expansive network of people who you can rely on. And essentially, and the idea of like extended or chosen family came up a couple of times. And it's especially pertinent for like the queer community because chosen family is often like what people end up having to rely on, because their biological families might not be as understanding of their identities. We also saw quite a few ideas. We saw like the theme of the passage of family heritage. So many participants considered passing on family legacy, heritage, or memory as a key factor to family building. A couple of people mentioned the realization of possibilities beyond the nuclear family. And while others even mentioned that essentially one of our participants in transition mentioned that they felt that they're at least becoming involved in like getting fertility treatment may allow them to become like blueprints for future people to have to become like to receive fertility treatment essentially. However, we did identify a couple of barriers as well. So namely, the first one basically being that there's an incredibly heteronormative care environment that causes like these clients or these patients a lot of emotional pain. So some examples of these are like highly gendered facilities. We had like one participant mentioning or relating an experience where they basically felt incredibly uncomfortable. So this was a trans man, and he was doing some egg donation, basically, or not egg donation, but egg collection, and had to do it in an all-female facility, which is suboptimal. Another example is that in certain like medical record keeping software, semen samples can't be attributed to female profiles, and this kind of denies a person in transition the ability to like transition within the existing systems. We also found certain examples of systematic exclusion of LGBTQ+ people within the industry, and I mean I've just left that on the slide up there. So that's a really quick summary of what went down, but I'm running out of time. So if you're interested in my work, hit me up. Email's there.
Thank you so much, Chloe. We appreciate it. It's like we got 2 lectures for the price of 1. And I think they're important for all of us here. I have a question on your first lecture. You talked a lot about this modeling. So tell me exactly, what do you hope to be able to use this modeling for? How's it going to increase our understanding of the embryo?
So effectively, what I want to look at is the actual dance of the blastomeres, so how blastomeres move around and essentially whether their positioning or like who they come in, which other blastomeres they come in contact with, has an impact of where they basically end up in the morula and in the blastocyst. So looking at very, very, very early like cell fate decisions, essentially, and doing this without confocal microscopy, and bringing this sort of research into the clinic rather than having it as the preserve of the academic lab.
Wow, that's great. I think a lot of us older people remember some of the earlier work that was done with making of geeps, which are goat sheep embryos, and that however old the embryo was determined which cell was going to become an inner cell mass or a trophectoderm cell. So I can see that this could be very important. I'd like to thank everyone that's taken part so far in our discussion here, and especially Chloe for a very interesting discussion. Now I think what we need to do is we need to get ready to have a Q&A section. Huey?
Yes, that was awesome, Chloe. I appreciate your thoughts. This question is pretty much for Chloe or Radhika, whatever. I'd like actually both your thoughts on it. With all this AI work that you guys are doing, which is amazing, and I think it was, like I said, of great benefit to the future of IVF. Where do you see the morula? Because in IVF we kind of ignore the morula. How do you see AI helping us potentially looking at maybe more insight to the morula and as a part of like helping us to grade and transfer compared to the blastocyst? Everything's about the blastocyst, right? But how can AI help with getting more insight on the morula?
Well, I'm sure Chloe can definitely expand on my very childlike opinions about this, but I completely agree. I think the morula is definitely neglected, and I think because we have been favoring blastocyst transfer for so long, we just kind of hoped for survival of the fittest, and we're just going to choose whichever one becomes the blastocyst. But the way I see AI transforming the way we look at the morula is looking at the finer details. From what I understand, using particular methodologies within AI that Chloe can explain a bit better, it's essentially going to look at it in a way where maybe we can classify movement and correlate it or associate it to blastocyst formation or fetal heartbeat post-transfer, something like that. That's where I intend to go, where I try to find associations between movement or pixelation or presence of vacuoles, anything we do have to kind of try and find any clues as to what we're looking at, because that's the biggest problem. We don't know what we're looking at, but maybe AI can. Maybe they can create a quantified method of grading. Maybe it's going to be qualitative, but we're going to have to try in order to find out.
And I'd like to remind everyone, not all of us have been hung up on blastocysts. Dr. June Tao in Chandler, Arizona, that for years has been transferring morula and feels it's a better stage for transfer of embryos. So, and I heard of I met someone, I think it was at the last ASRM, that mentioned to me that they also do morula transfers. So, there are those people that have not followed the dogma and that have been on the edge trying to learn new things.
Yeah, I feel like those people should be speaking up too, I think it's about creating that voice a little bit where you have to tell people that you do something different and they don't just get shot down because of it. We need to hear the different sides of things.
Yeah, let me remind everyone, this is the Q&A session. So anyone with questions, type them in the Q&A section. Don't put them in the chat. I have a question for Angela, one of our panelists. So Angela, one of my pet peeves is the, the signing of a contract for a clinic that then says that you can't work for anyone else in the area. And I know this is probably not just an issue in the United States, but it must be an issue in other parts of the world. These restrictive clauses that say you can come work here, but once you leave here for 1 year, you can't practice anywhere in this region. How do you feel about that? And do you think that's something that that we should just say, oh, well, it's going to be there, we should just handle it? Or is there something we can do?
Yeah. Thanks, Kim, and thanks for inviting me to participate this evening. So I'm based in the UK, and I think the way that in the UK these things are perceived and the way that they work in practice is that it's about proportionality. So if you work in an organisation where you have a job role that's very important, potentially very important from a commercial perspective or an intellectual property perspective, then those restrictive clauses are relevant. If you work in a role where you want to have options and be mobile, the courts in this country won't enforce those clauses. And it would be unreasonable for employers to expect employees not to be able to earn a living or to move. And I think the challenges for employers, we need to take action that makes people want to stay with our organization.
Right. These they're called non-compete clauses here in the US.
Yeah.
And I think we can get rid of them very quickly. And I think all you have to do is when you interview for a job, the first thing you say is, before we go any further, I want to know if there's a non-compete clause involved here. And then after a few times of them hearing that I'm done with this interview because of a compete clause, those non-compete clause will be gone.
Yeah. Yeah.
And
they're not
illegal in most
standard.
places, but then you have to fight them.
Yeah. Yeah. So certainly in the UK, they're not standard, only for those higher level roles where there is a real risk of somebody leaving an organization and going to work for a competitor. But in most roles in our sector, that doesn't apply.
Awesome. I have a question from the audience for Chloe. Let's see here. It's from Francesca Bear from Global Product Manager from Future Life. Can you analyze the 3D models and link characteristics of 3D versus 2D features that are uniquely correlated to clinical outcomes?
Yeah,
yeah.
I mean, that's the long-term goal.
Yeah.
So yeah, it's in the works at the moment. I'm looking at just static analysis of cleavage stage embryos. Yeah, we do have time-lapse, but analyzing time-lapse is hard and effort. So like, go for the low-hanging fruit and see whether or not it even works, and then go from there. We know, like from just standard clinical research with people looking down microscopes, myself included, that like in very early stages, we can find like links to clinical outcomes between cell arrangement, and yeah, but it's essentially just a matter of crunching the data, and like I'm one person in working on this, so please give me time.
No. I think you're taking a great first step, so I appreciate your research. So I'm definitely going to follow you in the future.
Yeah, and actually, if I might add something on the morula question, if people even remember that question, I think that like it's very much a issue of the imaging modality and like how much we're actually willing to put in. So if it's just people looking at the morula through a regular microscope, well, firstly, you're looking at a static observation because embryologists are busy people and they don't have time to like watch it blob around. But simultaneously, you've got a microscope that can't really look at the cell junctions because they've all basically dissolved away because or like become invisible to light microscopy just because of compaction. And I think that like there are two ways out of this. First, not look at it statically. So don't do a static analysis, analyze it over the whole time lapse or like over time. That might yield something interesting. And that's what AI is doing. On the other hand, what you could start looking at is what if we augmented the actual like microscope? So what if we started looking at, for example, hyperspectral imaging or like differential phase contrast, and these things are just very, I mean, that's a lot harder because you have to go through ethics and basically prove that none of these kill the embryos. But yeah, I think that the morula is just neglected because it's just a pain to work with.
Yeah.
Toyin, I've got a question for you. You're on the panel. Tell us a little bit about what your job is, what you do.
So, hi folks, I'm Toyin Ojo, and I'm a trainee embryologist at a clinic called Repromed Island in Galway. And I'm training to become a clinical/medical embryologist. So I graduated from my master's there this time last year in November in Dundee. So I studied biomedical sciences for my undergrad. And then I went into ART, so Assisted Reproductive Technology and Human Clinical Embryology at the University of Dundee. So yeah, I've always done well in biology from like school days. And then in my undergrad, I was really drawn towards reproductive medicine and genetics. And I had the opportunity to go and do an internship in Leiden in the Netherlands into pathology. And I met a few doctors there as well. Also, I just found it really interesting, and I was like, this is like a way to kind of create life, and it's helping folks who I'd say there's not much light shed on people who can't have children. We kind of take it for granted that it's a process that is natural, and I just found it very interesting, and I really wanted to dedicate my future into helping others in this role.
So what was your impetus for getting a master's degree instead of going right into human IVF?
So I studied in the United Kingdom. So there is like 2 different ways you can go into the IVF sector. So you can do something called the scientific training program called the STP through the NHS. And that requires you to do 3 years of training and also gain a master's, but it's highly competitive and they take about 1, 2, 3 students a year. So it's slim chance of you getting into it. Or you can get trained through a private clinic. Most private clinics in the UK would require you to get a master's, and others don't. Really depends. It's also competitive, helps you, like if you have a master's, it pushes you forward and getting into the trainee role. And also it was the degree in Dundee was, it had like the clinical side, it had the scientific side, and I felt like I would be more in depth into the field instead of like just getting a broad spectrum for my undergrad.
Okay, thanks.
Awesome. So I have a question for Savannah. Some of the issues and topics that we've heard today, what are your thoughts on how, as a Gen Z, someone like Angela can help you stay within the industry longer and make it something that you really enjoy and dedicate that for the rest of your career in IVF?
I guess that's like a very broad question.
Yeah.
I don't specialize in embryology and training as an embryologist, but I'm still very closely associated with the field and like through my PhD studies. And I guess it would just be a case of opportunities for young people. Just as Toyin said, it's very competitive to get into it. And coming out of COVID as well, everyone's getting back into things and opportunities opening up, and they're hard, but as long as those are available to the younger generation, and that there's education schemes just like these webinars where people are exposed to free education and new ideas that keeps everyone motivated, then I think that would be a really great way to incorporate younger scientists.
Awesome. And Angela, what are your thoughts on do you, as you look to I guess recruit young embryologists, what are some strategies that you use versus recruiting a senior embryologist? Do you apply them across the board, or do you consciously have 2 different strategies, even though, openly or not? Right?
Yeah, no, I don't think we have to consciously have 2 different strategies. The challenge is to meet everyone's needs and compete
Sure.
for talent. It's a really, really competitive field for us as employers as well. So, turning on its head, it's not just competitive to get into the field, it's then competitive for employers to keep the talent. So, when we look at the generation gap, is the gap real, or are we comparing different things? So the early career embryologists of today versus the early career embryologists of 20 years ago, it's a different world. And employers understand better now the needs of employees. 20 years ago, we didn't really understand how we could help our employees. It was arguably it was much more straightforward for HR 20 years ago. But now we are expected, and we want to meet our employees' needs in a whole host of ways. And like Savannah said, it's around providing lots of opportunities, lots of career development pathways, whether that be clinical, academic, leadership, providing opportunities for people to leave the organization and come back if they want to do something new and different, giving them opportunities to go overseas and come back. And just ensuring that we can understand their values and meet them. So, you know, going back to Fran's presentation, you know, it's so important that we understand the importance of sustainability, not just because that's what employees expect of us, but also our patients expect that of us as well. So it's about listening to what employees need, whatever generation they are, and doing our very best to meet those needs.
That's awesome. I love your perspective on that. That's just about what the employee needs, and not necessarily a generational thing. We focus on different generational gaps, and sometimes, you know, it's just about what the person needs and how you can make them feel content. So that's great.
Fran, it looks like you have landed.
I have indeed. Sorry if you,
Oh,
I hope you can hear me.
Yeah.
But yeah, we're just waiting for the suitcases to arrive.
That's
But
awesome.
this is a truly Gen Z conference. I've got a question for you, Fran.
Yeah.
I see a lot of the waste that you see also. And I don't know how we can, you know, I feel like, well, I can't use these styrofoam containers. I can't use some of this waste. I love your idea about weighing out the semen. Why didn't we think of that before? It's got to be a lot more accurate, knowing the weight of your cup before, and then just weigh the cup once it's collected.
Exactly. And I think this is what the newest WHO semen analysis manual has said, that for more accuracy, you're not losing sample. And in the long run, for clinics, if you want to speak financially, you're going to save money because you're not spending so much on serological pipettes. So it makes sense to be doing it. I know some people are doing it, but I think it needs to become the norm. It just seems logical. I don't know why we're not doing it.
It seems strange that when I started all this, we used tons of glassware, which of course was reusable, but we went through arduous amounts of water to clean this glassware and make sure that it didn't have, you know, any toxins on it. So that's why we've moved to this plasticware. So I know there's some parts that we're not going to be able to be as green as we want to be, but
For
what
sure,
other
yeah.
areas can you see that we could focus on being green in the lab?
Well, I think we all have different responsibilities. The suppliers in their packaging, they could have just, you know, make it easier for us to be more sustainable. In the lab, as another Green IVF webinar saying in the setup, if we can reduce the number of tubes or the number of dishes, not by impacting patient quality of care, but like if you've got 2 tubes holding half the volume, can you use 1 tube with double the volume? Like you have the same amount in the oven or in the incubator, but you're not using as much tubes. So kind of seeing where you can not cut corners, but just reduce what you're using. And then, I mean, there's loads of different things. In clinics themselves, you can be offering plastic-free sanitary towels or pads, or, you know, just trying to be less wasteful in that department. You really just have to wear a pair of green glasses and kind of look around and just be like, that is wasteful. Cups, if you've got disposable or plastic cups and you're giving them to patients for water, have reusable cups. There's really no reason why we have to have these rubbish throwing in the bin ones. All the suitcases are about to start.
Thank you very much, Fran.
Okay, so I need to jump in there. I never thought about, Kimball, have you heard anyone ever talk about weighing the sperm? Like, what about viscosity? Like, a viscosity would impact the weight, right? Versus a very thin, you don't think?
Yeah.
I
know that somewhere in the WHO manual, it's written, it has the density calculated. I just, a little bit of jet lag, don't know it on top of my head. It's definitely a thing that I've not just made up, it's already in action. And then if there is slight not inaccuracies, but differences with density. I'm sure you can calculate that out. But I think it's very similar. I don't think there is actually a density issue.
That's correct, Fran. I know that someone here has made a comment that said, alert Kimball Pomeroy to both the mushroom styrofoam alternative, as well as the possibility of replumbing of wastewater to laboratories, and maybe the other panelists also. So there are things that can be done. In some areas, there may be some building codes that have to be taken into consideration, but no one's going to do this rerouting of plumbing so that you use something like hand water to flush the toilets, which seems logical to me, unless someone stands up and says something about it.
I think the challenge of, Fran, I love your ideas of green solutions, green strategies in the IVF. You know, I would personally love to see that implemented more. But in the US, you know, I used to teach environmental science, and the challenge was always to get people to take that first step, right? And that's a challenge. You know, can you get in the US, you know, can you get the employer to do that? You know, you have to, unless we find a way to make these green strategies profitable or a reduction in costs, it's going to be challenged to get these, especially in the US, clinics to give a crap. You know, like,
they very often go hand in hand, though, don't they?
Yeah, they should. That's
they
why
do go hand in hand.
They should. You're right, Angela. And I think if you can make that hand by hand, I always tell a lot of my business students that are in my environmental science class, look, you know, sustainability is real about saving costs. So I think if you can really define, at least prioritize like maybe a handful of the top green solutions that you have, and try to find the ROI on them. Because if you do that, then I think you can get clinics to really implement them, and it'll be a better thing for all of us in our plan and everything.
I think it takes focus and commitment. And in my organisation, we're lucky enough to have a dedicated post to sustainability, that makes a difference, because there is somebody that has the time and resource to coordinate these things and educates everybody, and look for the opportunities, and tapping into lots of external resource actually that can help organizations do better with sustainability.
I also think by having this conversation, if you're planning on setting up a clinic or building a clinic, that's when you can start implementing those things. So maybe not, well, we can try for the ones that are already built, but the ones that are to be built, that's where we can really make some change as well. So just having that conversation is starting the journey, I guess.
Then that's great. That's a great point.
Deb Veneer in California mentions there's a styrofoam that's made with starch that dissolves. And I think I've seen that. I think I got something in the mail the other day, and my wife could actually just take it and put water on it and dissolved into nothing. It doesn't allow you to necessarily reuse it as well, but at least it doesn't take up the landfill as much. I want to ask a question of anyone here who would care to answer it. This is from Rusty Pool in the United States. He says he wants to know how people feel about regarding our branch of medicine as an industry. He finds it derogatory and can't think of another subspecialty. And you think about it, why do we often say the fertility industry? We don't say the brain surgery industry. Angela, do you want to take a stab?
Absolutely. It's a word that I don't use, and I don't believe my colleagues use. We use sector, and I think that sets the tone of what we do. I don't know what the other panelists view on that, but
I hear the word industry more in the the Northern and American side. I say over here in Ireland, UK, Europe side, it is called the sector, as Angela did say. I don't know if it's a thing in America, more of a money making scheme people would see than actually helping people. But we don't call it. I've not heard it called an industry over here. I have heard folks who live in America who are in clinics, they call it the industry. I don't know why it's called the industry. It would be nice to find out why, but it is, I do feel it is derogatory.
I wonder if it's similar to how we apply the term embryologist or technician or technologist. I know when I go to Australia, they're called scientists. So Are we now becoming less scientists and more technologists? What's happening with our industry? Industry. It's an American thing. What do you think, Savannah? Do you think you can kind of look at it from the outside? I don't know if you've seen these changes or the application of these terms. Are we less technology driven and more recipe driven now in IVF?
Well, I think that even if it were to be more technology driven, then that wouldn't discredit from being a sector or a field of science that we can provide a service for people and improve the fertility journey of people around the world. So regardless of what you'd call it, the movement of the science, whether we're using technology or old methods, yeah.
I agree with you, Savannah. I don't get hung up on these terms. These terms are whatever
I don't, yeah.
you apply to them. So technologist could be a derogatory term, or it could be a very good term about someone that understands the technology.
Yeah, I mean, just from what Chloe's work is and Radhika's as well, incorporating technology and modern aspects of data science and computer science, that's far out of my reach. And that would, in my eyes, still be a major contribution towards our sector, our industry, our field, whatever you'd like to be called. Yeah, I agree.
Thank you.
I do believe technology is the way moving forward. So we do need to incorporate it at some stage, and it's for the better. For example, what a human eye can't see, the computer can see. For example, we use the embryo scope for time lapsing, saves a lot of time. That's moving forward. And there'll definitely be better time lapse machines in the future, which will, as Chloe did say, and it comes to, like, let's say, looking at the morula, like, how can we make that better? Like, so we do have to use technology moving forward.
Yeah, I think we'll always be some sort of technologist. We probably pride ourselves on following protocols or recipes, whatever we want to call them. We have to have pride in that so that we decrease variability. But we also have to have pride that we understand how these things work and the intricacies. So when there's a problem, we can solve an issue and solve it creatively that will be to the benefit of our patients and not damage their tissue.
Awesome. Yep. I have a question from Kimball. I don't want to interrupt you.
No, go ahead.
I had a question for Radhika from actually one of the audience from Phil Chang. I thought it was a really good question. I wanted to have your thoughts on, Radhika.
Mm-hmm.
In moving from one lab to another, you encounter different protocols, freezing cells, etc. Do you find educating the new lab staff to change works better, or do you just follow their protocol that has been working for years? You know, as a millennial or as a Gen Z, like as you're entering a new lab, when do you make that transition into expressing your thoughts?
Yeah, that's a great question. And to put it really plainly, in my first experience as an embryologist, I did not question anything. I was too scared. Yeah. But I think it depends on the relationship that you have with your team. And more than that, I think change is taken up by people in your team a lot easier when everyone understands why the change is being made. So if a change is just happening just because, oh, you know, there's a new product and we're just switching, or, you know, if we were using derogatory terms before, if someone from the industry came for some sort of demonstration, now we're just taking it on, that change would be it would be assimilated slower or less frequently than if everyone understood how the difference was being made, or whether there was actually a difference between the existing protocol and the future protocol. So I think I'm still a bit early in my career to assert that much change. I haven't reached that level, but I hope that the conversation would be easy to have.
Yeah, I would hope that anyone that's been in the lab a while would listen to advice from someone that's come from someone else. I know I always appreciate it. When I do inspections, for example, in the US, we have what are called CAP inspections. I learn more when I go and do one of those. And I would hope that someone would always appreciate that. But I think we need to close this session up. And I'd like to thank everyone who took part in the webinar. So Huey, I'll let you take it over there.
Okay. So I'd like to thank everyone. I actually learned a lot from this webinar, from hearing, just hearing and seeing the excitement of the panelists and the speakers here. It's really just kind of rejuvenating. It really made me think about other things that we can do to improve our field. But I'd like to thank everyone who took part in this webinar, of course, my co-host, Dr. Kimball Pomeroy, our panelists, Savannah and Toyin and Angela, and our presenters, Fran, Radhika, and Chloe. This has just been a great talk. And of course, I don't want to forget the team from I3. Thanks for organizing the committee for this initiative. I3, who are Jacques Cohen, Pierre Naj, Marianne Sebtez, Victoria, and Thomas Ellie, of course, for technically making sure that we're all on par. And of course, Giles, and most importantly, Fran, are the true ambassador of I3 ambassador. So thank you so much.
And thanks everyone for joining us. And I hope you enjoyed this session. Look out for the podcast that we have that accompany this session. This session will be on the IVF I3 website. I3's next session is all about the highlights of the 13th Biennial Alpha Congress, in case you missed it. On December 6th of this year, there will be award-winning talks and presentations from the event in Seville, with news updates from Alpha, including the meetings in the future and a competition also. So stay tuned, and we look forward to seeing you on I3 in the future. Thank you, everyone.